release free energy, which means that delta G is negative, and endergonic reactions store free energy, which means that delta G is positive. Pretty confident in this one, that's good. Some things that I want you to know about this. So not only does delta G being negative mean that energy is being released, but also it means that it is spontaneous, meaning it can happen at current conditions, right? If delta G is positive, that means it's non-spontaneous, so it's going to need some extra energy input in order to get it to happen, right? Normally when we talk about the release of energy, you hear the term exothermic, right? But exothermic refers to something slightly different because we wouldn't talk about it with delta G, we would talk about it with delta H, right? Delta H is the change in enthalpy, right? And that would be what would determine whether or not something is endothermic or exothermic, okay? Topic 2 says, ATP powers cellular work by coupling exergonic reactions to endergonic reactions. How do you feel about that? Decently confident. Basically, the hydrolysis of ATP is exergonic, meaning it's going to release energy. If you have an endergonic reaction, you need energy from somewhere. Where do you get the energy from? You get it from the breakdown of ATP. ATP becomes sort of like this universal energy currency then that can be used to power all endergonic reactions, or power most endergonic reactions. Topic 3 says enzymes work by lowering the energy of activation. Feeling pretty confident in this. Energy of activation or activation energy is the amount of energy required for a reaction to get started. So we got on the potential energy graph, you get that hump that you got to get over, right? And so by adding an enzyme, that lowers the hump, which means that you don't have to have as much energy. This is important because really the only other way to get the reaction started would be to increase the temperature. And we know we don't want to do that in biological systems. We want to keep a relatively constant temperature and then allow for the enzymes to reduce the activation energy. Topic 4 says the catalytic cycle of an enzyme that results in the production of a final product. All right, so we definitely need a refresher on this one. So I think most people are confused just because of this catalytic cycle part here. That just means how an enzyme works. So we know that an enzyme has an active site, and then there's a substrate that fits into that active site, right? So substrate has like a lock and key fit with the active site, and then the active site will do one of two things, right? It can either ensure proper orientation between two or more substrates, right, to allow for a reaction to happen, or it could be a single substrate where it will then put a strain on it. It will cause it to be easier to break the bonds. We call this the induced fit model. So if it's a single substrate that fits in there, you got the induced fit model, and then it'll have some reaction site that's exposed, or if there's multiple different reactants that can fit into it, then it can ensure proper orientation. And both of those will end up reducing the activation energy and increasing the reaction rate. Enzymes are specific in the reactions they catalyze because the molecular shape of their active site. Highly confident in this one. We just know that every enzyme has some substrate that fits into the active site. That's great. Topic 6 says factors that change the shape of the active site and how they influence enzyme activity. All right, so we're less confident in this one. There's two different ways that the active site can change, right? The first way is on purpose, right? So we've got enzymes that have an active site, and then away from the active site, they've got the allosteric binding site. And as something binds to that allosteric site, the active site can change its shape. And so we say that if it changes to an active form, we call it allosteric activation. If it changes to an inactive form, we call it allosteric inhibition, right? That's the on-purpose change of the active site. Then you've got the more random changes to the active site that can occur as a result of denaturation. So denaturation occurs via changes in temperature or changes in pH, and we know that that is ultimately going to decrease the activity of the enzyme, right? Because it's going to make it less effective. It's going to fit less of the substrates. Topic 7, how feedback inhibition is used to maintain appropriate levels of enzymes and enzyme products in a pathway. Again, we need a refresher, it looks like. So, two types of feedback inhibition for enzymes. You've got competitive inhibition and non-competitive inhibition. Competitive inhibition means that the product of the reactant, or product of the reaction rather, is going to come back and it's going to bind to the active site and prevent further substrates from getting into the active site. That's going to lower the activity of the enzyme, which will then lower the reaction rate. The second way is for the product of the reaction to come back and allosterically bind to the allosteric site to change the shape of the active site and prevent more reactions from occurring. Both of them will prevent further reactions from occurring, right, which then is going to maintain homeostasis, right, but just it's two different types. So allosteric or non-competitive versus competitive. And then you can also have a positive feedback loop that wouldn't be feedback inhibition, right? But if a positive feedback loop would be like if the product of a reaction came back and bound to a allosteric binding site, which then would activate more of that enzyme, right? And that would make more products happen, which would then activate more enzymes, and more products happen would activate more enzymes, and that would not maintain homeostasis, but that would be something that was called a positive feedback loop. The summary equation of cellular respiration, including the source and fate of the reactants and products. So the summary equation is, for cellular respiration, is glucose, which is C6H12O6, and oxygen, 6O2, yields 6 CO2 plus 6 H2O. And we know that when we talk about the fate of those reactants and products, the C6H12O6 is going to be used to create all the carbon dioxide, and then the water that's the product is going to be created as a result of oxygen picking up those terminal electrons, right? So at the end of the electron transport chain, oxygen accepts the electrons, and then that allows for the water to be made. Topic 2, the difference between fermentation and cellular respiration. Pretty confident in this one. So fermentation is anaerobic, meaning no oxygen needed. Cellular respiration is aerobic, meaning it requires oxygen. Fermentation is going to be only glycolysis versus cellular respiration. It's going to be glycolysis followed by the Krebs cycle, followed by the electron transport chain. Fermentation produces 2 ATP per molecule of glucose. Cellular respiration produces 36 to 38 ATP per molecule of glucose. Fermentation also results in the production of either lactic acid or alcohol, which is a toxic byproduct, which if left unchecked will kill the organism, right? So the buildup of too much lactic acid in your body would kill you. The buildup of too much alcohol in the environment of, say, yeast will kill them, right? It has to be dealt with, otherwise you will die. Topic 3, the role of glycolysis in oxidizing glucose to 2 molecules of pyruvate while releasing free energy to form ATP. Need a refresher for sure. Okay, so I don't really know what they want you to know about this. It's kind of just the thing, right? So glycolysis takes 1 molecule of glucose and breaks it down into 2 molecules of pyruvate. How does it do that? Well, it's going to first need to invest some ATP, right? So it's going to spend 2 ATP, and then it's going to get 4 ATP back, and that's going to result in a net of 2 ATP, and it's also going to produce 2 NADH, which would then be used in the electron transport chain. How pyruvate is moved from the cytosol into the mitochondria and then introduced into the citric acid cycle. Lots of never even heard of this. And that's fine because we didn't use these terms very often, right? So let's start with this one, cytosol, right? So cytosol, we didn't really talk much about cytosol in here because the term that we use is cytoplasm. There's, in a cell, you've got this space that is in between the nuclear envelope and the cell membrane, right? The space between the nuclear envelope and the cell membrane is called the cytoplasm. The cytoplasm is filled with a solution, with a watery-like solution, that is called cytosol, right? So by saying that it's moved from the cytosol, really they're saying it's moved from the cytoplasm, and then into the mitochondria, right? So how does pyruvate move from the cytosol into the mitochondria? Well, in the mitochondria, you have a really high concentration of pyruvate versus the cytosol. So how do you move something from a low concentration into a high concentration? You have to use active transport, right? So this is going to require active transport, and specifically this active transport is going to use ATP. And when we talk about that discrepancy between eukaryotic and prokaryotic cellular respiration, that like we say that you make between 36 and 38 ATP, right? The 36 to 38 has to do with the 2 molecules of pyruvate that have to be actively transported into the mitochondria in a eukaryote, but not in a prokaryote. What's up?
Sort of like the citric acid cycle.
So that's the other thing that we didn't talk about, or that we didn't, the terminology that we didn't use in here, and that is the citric acid cycle is just the Krebs cycle, right? So the citric acid cycle is called, or the Krebs cycle is sometimes called the citric acid cycle because the very first molecule that's produced is citrate, and when citrate mixes with water, it becomes citric acid. In biology or all sciences, after a certain amount of time of naming something using a person's name, so Krebs, Hans Krebs was the guy that discovered the Krebs cycle and really came up with most of the stuff in it. For some reason, they always, after X number of years, start moving away from that name, and so they're calling it the citric acid cycle now to kind of remove it from Hans Krebs, because his name, even though he discovered it, it doesn't have anything to do with the cycle, versus the citric acid cycle has everything to do with it because that's the very first thing in the cycle. So how does it get incorporated into there? We know that pyruvate goes through the Krebs steps. It gets turned into an acetyl group, and then as the acetyl group mixes up with, or gets bonded to rather, coenzyme A, then that acetyl coenzyme A becomes the very first entry molecule into the Krebs cycle, or into the citric acid cycle in this case. Topic 5, which is how electrons from NADH and FADH2 are passed to a series of electron acceptors to produce ATP by chemiosmosis. Need a refresher. All right, so NADH carries high energy electrons. FADH2 carries slightly lower energy electrons. And then they all get passed into the electron transport chain. The electron transport chain contains these protein complexes that we call ferredoxins and cytochromes. And basically, the ferredoxins and the cytochromes, their secret sauce is that they have metals in them, right? They contain iron. And so because they contain metals, they can accept electrons, right, of certain energy levels, and then they can give away electrons of certain energy levels, right? They can change their oxidation states. And so what happens is it gets passed in at a really high energy level, and then it gets passed down from one ferredoxin to the next ferredoxin to the next cytochrome, yada yada, right? Until it gets to the very end where that electron is now used up all of its energy. And every time it gets passed down, it's used to pump H+ ions, right? So that ends up with more H+ ions in the intramembrane space. And then when it gets to the very end, and it's kind of this low energy electron, the only thing that can accept it, or the only thing that wants to accept it, is the oxygen. And oxygen is super electronegative, so it's super good at yanking that low energy electron away from the last ferredoxin, so that it can then form water, and then the process can continue. Topic 6, which is the roles of the mitochondrial membrane, proton gradient, and ATP synthase in generating ATP. Again, looks like we need a refresher. So this proton gradient is created by this, right? Proton gradient gets made inside of the intermembrane space. So H+ ions are pumped from the mitochondrial matrix into the intramembrane space, which means there's a high concentration of H+ ions in the intramembrane space. And the only place that they can flow out is through ATP synthase. So they flow out through facilitated diffusion through ATP synthase, and then the energy of them flowing down their concentration gradient is captured in order to produce ATP. Yeah?
The gradient, is this the one that's created by the superconductor?
No, that's in photosynthesis. Okay. So this gradient is created through the electron transport chain. Okay. All right. So now the last piece here is this one. We'll start with that. So the summary equation of photosynthesis, including the source and fate of the reactants and products. Short refresher needed. So summary equation, in photosynthesis you need carbon dioxide and you need water. So 6CO2 plus 6H2O, and it makes glucose, C6H12O6, and then it makes oxygen as a byproduct, so 6O2. So the source and fate of the reactants and products, we know that the water is going to get broken down in the light reactions. The very first thing that happens in the light reactions is photosystem 2 absorbs light and uses that light in order to break apart water in a process that's called photolysis. So the oxygen comes from the water, and then the glucose comes from the CO2, meaning that in the Calvin cycle, what happens is that the very first step is that this molecule RUBP is going to meet up with a CO2 molecule, and then CO2 is going to get added on to the RUBP to get everything rolling. Topic 2 says how leaf and chloroplast anatomy relate to photosynthesis. So people are pretty confident in this. Basically, we know that photosynthesis requires light, and so you want to put the chloroplast, which is where the photosynthesis happens, you want to put them pretty close to the surface of the leaf where the light is actually going to hit. And so you've got a thin layer called the epidermis on a leaf, and then under the epidermis, you've got this large layer that's called the palisade mesophyll, and that's where you're going to have the highest concentration of chloroplasts is in palisade mesophyll cells, because that's where all the light hits, and the light is needed for photosynthesis, and so then that's where you put them. As far as the chloroplast goes, we know that the thylakoids are where photosynthesis is actually taking place. So the light reactions happens in that thylakoid membrane, right? And then the Calvin cycle happens in the stroma, which is outside of that. Topic 3, which is how photosystems convert solar energy to chemical energy. Definitely need a refresher. So, what is the molecule type called? The type of molecule that converts solar energy to chemical energy. We would call that type of molecule that converts solar energy into chemical energy. There are many types of it. It's called a pigment, right? So a pigment is a specific molecule that converts solar energy into chemical energy. How does it do that? Well, it's got a similar secret sauce to the ferredoxins, right? Inside of chlorophyll is a magnesium ion, right? And that magnesium ion can change oxidation states. And what it basically does when that happens is when a chloroplast, or when a chlorophyll molecule rather gets hit by high enough energy photons, it sheds that electron, right? It gets rid of that electron, and that electron is now a high energy electron that can then be captured by the photosystem to aid in that process of photosynthesis. Light reactions, rather. How linear electron flow in the light reactions results in the formation of ATP, NADPH, and O2. What did we call linear electron flow in our class? We didn't call it linear electron flow. What's that? Non-cyclic flow. Yeah. So you got two types. You got cyclic electron flow like this, right? And then you got non-cyclic like this. So this is the linear type, right? And so in the non-cyclic flow, you've got ATP, you've got NADPH, and you've got O2. That's O2 is created as a result of splitting water, right? So your H2O becomes O, and then that happens twice, and then you get an O2, right? Your ATP is created as a result of the H plus ion gradient that's created not only because water is being split, but also because H plus ions are being pumped. And then those electrons eventually end up in an enzyme at the end that's called NADP reductase. And NADP reductase is going to then add 2 electrons to NADP, which turns it into NADPH. How the formation of a proton gradient in the light reactions is used to form ATP from ADP plus inorganic phosphate by ATP synthase. So where in the chloroplast does the do the H plus ions build up? Thylakoid space. So they build up in the thylakoid space. That means there's a really high concentration in the thylakoid space, and then they flow out through ATP synthase. And as they flow out through ATP synthase, that energy is then captured to create ATP. Last one says how the Calvin cycle uses the energy molecules from the light reactions, ATP and NADPH, to produce carbohydrates, which are G3P. So basically, everybody needs a refresher on this one. And in the Calvin cycle, the very first step, you've got a molecule called RUBP. Then you got a molecule called CO2, right? They both meet up in an enzyme that's called ribisco, which is ribulose bisphosphate carboxylase, and it adds this molecule to this molecule, creating a 6-carbon chain. That 6-carbon chain, fructose 1,6-bisphosphate, breaks down into 2 molecules, right? 2 3-carbon chains. Those 3-carbon chains are called PGA. They each get phosphorylated using ATP. So they get a phosphate group added onto them. Now they're called BPG, right? BPG is then reduced, it gets electrons added to it by NADPH, right? And when BPG gets reduced by NADPH, it becomes G3P, right? So that process creates 2 G3Ps, essentially, per Calvin cycle, right? So you need to go through the Calvin cycle 3 times then to create 6 G3Ps, right? If you create 6 G3Ps, 5 of them will be used to regenerate all the RUBPs, and then 1 of them becomes a product of that process. So you end up with 1 molecule of G3P. A single molecule of G3P is only 3 carbons. If you wanted to make a glucose, you would need 2 molecules of G3P, which means you would need to go through the Calvin cycle a total of 6 times. What's up?
We need to know like the inner workings of the Calvin cycle, or we just mainly need to know like reactants and products.
Yep, reactants and products. Questions on any of that stuff? Your homework is 92 through 100. I will not see you for quite a while. I won't see you till Wednesday next week because Monday is a day off, Tuesday is SAT day, and then Wednesday. So be ready for that quiz, 92 through 100 by Wednesday.